Our next speaker is Dr. Valerie Soza. She is currently affiliated with Evolve Scientific Editing, but is going to be telling us about some research that she has done in association with being part of Dr. Ben Hall's lab, where the Rhododendron williamsianum genome was sequenced. So today, Dr. Soza is going to be telling us about gene duplication and differential expression of flower symmetry candidate genes in rhododendron. So take it away, Dr. Soza.
Thanks, Juliana. Let me see if I can share my screen. Okay, let me know if you all cannot see or hear me. Thanks for everyone being here today. I'm excited to tell you about some work we just recently completed. As Juliana mentioned, that was part of my work as a research scientist at the University of Washington in Ben Hall's lab. So we're going to switch now from the roots up to the top of the plants to talk about flowers. And as you all know, rhododendrons also have a huge amount of variation in their floral shape, as you can see from some of these images of rhododendrons. One component of shape is the symmetry of a flower. And there's two types of symmetry that have been described for flowering plants. Those are radial symmetry and bilateral symmetry. Radial symmetry has more than one plane of symmetry through the flower, whereas bilateral symmetry has only one plane of symmetry through the flower. So some examples in rhododendron of these two types of symmetry. Normally rhododendrons have 5 petals, and I've labeled them here in this panel A. Usually the upper petal I'll be referring to as the dorsal petal. And then there's 2 lateral petals and 2 ventral petals. And so in this case here in A, this is a really good example of a bilaterally symmetric corolla in rhododendron, where you only have one plane of symmetry here. In some cases in rhododendrons, you see an increase in petal number. So an example of that is in C here, where you actually, often when this happens, you get a radially symmetric flower that has multiple planes of symmetry. So you could draw multiple planes where the flower is symmetrical on either side of those. And not only does the corolla contribute to symmetry differences, but also in rhododendron, I think a really cool thing about it is that the stamens and the style also contribute to symmetry differences in rhododendron. And here's an example in F where the upper stamens are shorter than the longer stamens. In addition, there's curvature to the stamens and the style. So this creates a form of bilateral symmetry in rhododendrons. And we also see differences if you look at the side view of the flower. In some cases, there are flowers that are very tubular and they can be curved along the lower part of the flower or along the upper part of the flower. And this also creates a form of bilateral symmetry in rhododendron. So throughout the talk, I'm going to be using green circles to denote radial symmetry and blue triangles to denote bilateral symmetry. And flower symmetry is very variable across flowering plants as a whole. This is an evolutionary tree of some of the main orders in flowering plants. And just to highlight that the ancestral flower was likely a radially symmetric flower for angiosperms. And you can see by the red stars, all of these orders have evolved bilateral symmetry within them. And it's been estimated across flowering plants that bilateral symmetry has evolved over 130 times. And this is often thought to have evolved to facilitate pollination. So here's some examples here of bilaterally symmetric flowers facilitating pollination. And the top example is a bird pollinated flower where the stamens are positioned in the upper part of the flower depositing pollen onto the back of a bird's head. And in the lower panel is another type of bilaterally symmetric flower that in this case is facilitating bee pollination by the lower lip of the flower, forming a landing platform for the bee. And then I want to point out here on this evolutionary tree that rhododendrons are in this order Ericales, which is an early diverging lineage of this larger group here called the asterids. And I'm going to be talking about a model plant that occurs in the asterids that's been used to identify genes involved in flower symmetry. And this model plant is snapdragon, which occurs in the Laniales here, which is a later diverging lineage of asterids. And in snapdragon, it's a model plant. It's amenable to knocking down genes and creating mutants and figuring out what these genes' functions are in the plant. And by doing this, previous research has indicated that there's 3 main genes involved in controlling flower symmetry in snapdragon. Normally in a wild type snapdragon flower, it's bilaterally symmetric. It has these 2 dorsal petals, 2 lateral petals, and a ventral petal. And these petals are all a little different in shape. And when you look at this in a cross section, you see the 2 dorsal petals up here, the 2 lateral petals, and then the ventral petal. And by knocking these genes down at a time, what we've discovered is that there's one gene, cycloidea, which is duplicated in Antirrhinum. And so there's actually another copy of it called dichotoma. And these 2 genes are turned on in the upper part of the flower in the 2 dorsal petals. And they turn on another gene called radialis, which is turned on in the dorsal petals and then also in the lateral petals. So radialis is what contributes to creating these lateral types of petals. Cycloidea and dichotoma create these different types of dorsal petals. And then divaricata is the 3rd gene involved. This is inhibited by radialis. And so it's only expressed in this ventral petal. And that's what gives this ventral petal its particular shape. And so when you knock out cycloidea and dichotoma, then they no longer turn radialis on, and radialis is no longer expressed in the lateral petals. And then divaricata can expand across all of the petals and giving you a flower that just has all petals looking like the ventral petal and in effect creating a radially symmetric flower. So these 3 genes have been identified as responsible for creating bilaterally and radially symmetric flowers in snapdragon. And this has also been looked at, these 3 genes have also been looked at across other asterids to see whether or not they are acting similarly. And we do see some conserved patterns. So the 3 genes are indicated when they're turned on in 3 different colors. And across asterids, you can get radially symmetric flowers, basically 2 ways. You can, as I showed earlier in snapdragon, if there's no expression of cycloidea throughout the petals, then you do not get any expression of radialis throughout the petals. And then divaricata can expand across all of the petals, making them similar looking and creating a radially symmetric flower. Another way of getting a radially symmetric flower that has been observed in other asterids is that you can also have the cycloidea gene turned on across all of the petals, which will then turn radialis on across all of the petals. And this will inhibit divaricata, reducing its expression across the petals. And so you get a radially symmetric flower also this way. Another thing that has been noticed when we expand and look more at other asterids is that we often see when there's a shift from a radially symmetric flower to a bilaterally symmetric flower, we see that there's associated with that a duplication in these cycloidea genes, where we then have two copies of this gene, and their expression patterns change a little bit, where one of them becomes more restricted to the dorsal petals. And so this creates a bilaterally symmetric petal with dorsal petals that are different from the lateral and from the ventral petal. And so we wanted to know whether or not these three genes were also involved in the changes of flower symmetry we see in rhododendrons, and whether or not we see duplications in these genes associated with changes in symmetry in rhododendron. So we predicted, because we see variation in symmetry in rhododendron, we predicted that we would see duplications in the cycloidea genes and also losses in these genes associated with transitions to radially symmetric flowers. And then because cycloidea interacts with radialis and divaricata, we also predicted that we would see duplications and losses in these genes as well in rhododendron. And then we wanted to look at whether or not the copies of from each of these three genes, if present, had evolved different expression patterns across the flower organs in radially versus bilaterally symmetric species. So we wanted to look at expression patterns between two closely related species that had different flower symmetries. And we chose two species in the tropical group section Schistanthe. This project was initiated with a Chinese graduate student intern in our lab from South China Normal University. And we selected these two species, Bayerinkianum and Taxifolium. Bayerinkianum represents bilateral symmetry because if you look at the side view, its corolla tube is curved. And this curvature is due to the petals that are dissected below being different lengths. So you have the uppermost petal, the dorsal petal, the longest, then the lateral petals, and then the ventral petals on the lower part of the flower being the shortest. We also see differences in lengths of the stamen, which is kind of different from what you normally see in the temperate species, where in the upper part of the flower, we see longer stamens, and in the lower part of the flower, the lower stamens, we see shorter stamens. And then we also notice the style curves. In Taxifolium, this is more of a radially symmetric flower because there's no curvature to the tube in the side view, but also all of the petals are more or less similar in length and shape. And the stamens are more or less similar in length. They alternate in length. And the pistil is relatively, the style is relatively straight and short. So we wanted to look at expression patterns of these three genes in these two species and compare them across these different organs, the petals, stamens, and the pistils. But to do that, we first had to identify how many copies of these three genes we have in these two species. We also wanted to get a sense of the history of these three genes across the genus, whether or not there had been many duplications or losses in these genes. And so we first started with sampling across the genus. On the left here shows you a evolutionary tree showing you the relationships between 8 species of rhododendron that we sampled. And these are from 4 of the subgenera in the genus, subgenus Rhododendron, Hymenanthes, Azaleastrum, and Theriodium. And we isolated cycloidea genes from these 8 species. And this was done, initially started by an undergraduate student in our lab, Haley Deal. And what I want to point out here are these things here in bold are rhododendron cycloideas. And we did observe some duplications in these genes that happened independently in one particular species. So for example, Kamchaticum had 3 copies of this gene. But then we also noticed that there were some main duplications that happened that were shared by the other three subgenera, Azaleastrum, Hymenanthes, and Rhododendron. So there were three main duplications shared by these three subgenera, and then another duplication that was shared by subgenus Rhododendron. And these four main duplications led rise to 5 copies shown here in the blue triangles that we found in Bayeringianum, and 4 copies shown by the green circles that we found in Taxifolium. So in our 2 experimental species, it looked like there were 5 copies where one of them had been lost in Taxifolium. And then when we looked at radialis, this was work done by an undergraduate student in our lab, Videhi. This had a different pattern to cycloidea in that we found 2 copies of this gene in Rhododendron, as indicated by the blue and the green circles in our two experimental species. And these 2 copies resulted from a very old duplication that happened, or that was shared in an ancestor of the Ericales. We found these 2 copies also in kiwi fruit, which is shown here as actinidia. So this was a much older duplication than what we observed in this with the cycloideas. And we didn't find any subsequent duplications or losses in rhododendron in this particular gene. We found a similar result with divaricata, the third potential flower symmetry gene, in that we found 2 copies of rhododendron indicated here by the triangle and circle. And these 2 copies in rhododendron originated from another older duplication that happened in these genes that was also shared with other Ericales members. And we didn't find any additional duplications or losses in divaricata and rhododendron. And so now I'm going to show you, now that we know how many copies of each of these genes we have in the 2 experimental species, I'm going to show you some gene expression patterns that we observed across these different copies. And what I'm showing you here is a cross-section of the flower. And the pink color, the intensity of pink represents the expression level of these genes, like how much they were turned on. And so I'm showing you here the petals, the stamens, and the pistil. And what I first want you to notice is that across the 5 cycloidea copies, we see different expression patterns across these copies within one species. So for example, in one copy, we see a little bit of expression in the dorsal petal, but then in another copy, we see expression in the dorsal and lateral petals, in the upper stamens, and the pistil. And when you look at these copies in the bilaterally symmetric species, what we do observe is that we do see this conserved pattern where the cycloidea copies are being more or less, their expression is being restricted to the upper part of the flower, where they're turned on more in the upper part of the flower. And we see this not only in the petals, in the dorsal and lateral petals in some cases, but then also in the upper stamens. And this is also where we see longer organs. So we see these longer petals and longer stamens where these cycloidea genes are being turned on in the flower. And then in the radially symmetric species, there was a loss of one gene, but across the 4 copies in this species, we see, again, differences in expression among the copies where one is expressed just in the dorsal and lateral petals, whereas another one is expressed throughout all the organs. But overall, we see, again, there's some conserved patterns that we saw in asterids where we see more of an expansion of cycloidea across all of the petals and all of the stamens and the pistil. And so this expansion or uniform expression across all these organs is associated with radial symmetry, as has been seen in other asterids. Where we start seeing differences in rhododendron is when we look at the copies of radialis and divaricata. So if you recall in snapdragon, in a bilaterally symmetric species, radialis is expressed only in the upper part of the flower. But when we look at radialis in a bilateral and a radial flower of rhododendron, we see expression across all of the petals, all of the stamens, and the pistil. So expression of this in both of these types of symmetries is not related to how the flowers are looking in their symmetry. Similarly, with divaricata, in a bilaterally symmetric flower like snapdragon, we see it restricted to the lower part of the flower. But in rhododendron, again, we don't really see any differences between a radially and a bilaterally symmetric flower. We see that div is expressed across all of the floral organs in both types of flowers. So this indicates to us that radialis is not being regulated by cycloidea in rhododendron, and that divaricata is not being regulated by radialis in rhododendron. So what we've discovered is that sect is the main key flower symmetry gene in rhododendron. And we believe this because the expression of rad and div is not affected by sect expression. And so we believe that rad and div became evolved later on in asterids with regards to flower symmetry, and are not players in rhododendron in determining flower symmetry. And then I'd like to thank members of the Hall Lab and the Distilia Lab from the University of Washington for all of their work in this project, and for the Rhododendron Species Botanic Garden and Royal Botanic Garden in Edinburgh for allowing us to sample a variety of species from them. Thank you.
Thank you, Dr. Soza. Excellent talk, very fascinating. Don't forget to put your questions in the chat. And I have a question. So this seems very complicated. Is this more complicated than other plants, or are we just lucky winners of rhodos?
I think rhododendron is a lot more complicated.
Yeah.
Than other plants when you start looking at them at the molecular level.
Yeah. Those copies, those gene copies. Now I'm gonna put in the chat. Valerie gave a great podcast interview a couple of weeks ago, so I'm gonna put in the chat the link to that podcast interview, because basically in it you talk a lot about gene duplication in rhododendron in general. Do you want to give just a short synopsis of overall what you found on gene duplication?
Yeah, let's see. So I guess in rhododendron in general, there's at least evidence for two whole genome duplications that have occurred in the group that happened many years ago, millions of years ago, like 80 plus millions of years ago. So that creates, whenever the whole genome duplicates, that creates copies of the genes in the genome. So we have that with rhododendron, but then we also, another paper just came out, and what we found with cycloidea is that the radialis and divaricata genes, the two copies that we see, look like they're the result from one of these old ancient whole genome duplications. But the cycloidea genes, they're duplicating in rhododendron right now. And so they're creating a lot of copies, and they're all on one chromosome right next to each other. So they're duplicating in these like a hotspot, like recombination regions. And so I think because they're located in this region, you see this expansion of these gene copies in rhododendron that happens independently multiple times in different species. So there's a lot going on. There's a lot of gene duplication that has happened in rhododendron historically, way historically, and more recently historically.
And one of the things that you go into in the podcast is this is the beauty of having the whole genome sequence, is that you're able to say, hey, here's the cause of that duplication in the ancient history. Yes.
Yeah, so because we have a genome. So one of the useful things was that we had this Williamsonian genome. We don't have the genome of the tropical species sequenced, and we had to fish those genes out of the tropical species without a genome, but we can like map them to or use like the Williamsonian genome to figure out where these genes might be located on chromosomes based on the Williamsonian genome. So that's been really helpful. I forgot what was there.
That's okay. I think everybody's got a teaser. Now they can go listen to your podcast episode, because it was very, very fun. And she goes through all of this and the process. So round of applause. Thank you very much. Great talk. Thank you so much.